All right, so this lecture is a lecture on the physiology of the nervous system. And this exam will be on proctorio. And so it's not going to be like it's scheduled like on Wednesday. I know I'll be finished by Wednesday, so that's when I put the exam there. But I'll make the exam on proctorio available early next week. So I'll give you this week to study, and then you can take it on proctorio next week.
No, with proctorio, I thought it was. Okay.
So my opening slide is a sagittal view of the whole body with the nervous system highlighted. I'll start with kind of a bit of the anatomy here. It points to the cranial part and then the spinal cord of the central nervous system, or CNS as it's called. Basically, brain stem, spinal cord. Okay, anatomically, that's what it is in a nutshell. I have a whole lecture on each of these things, so I won't really get into it. We have a cranial part and then a spinal part, and that's the CNS, anatomically. Now, for the CNS to control the whole body, it needs a complex system of nerves. So what we see in the peripheral nervous system, if you were to remove the brain and the spinal cord from their bony cavities, remove the brain from the skull, the brain stem, and then all the way down to about L2, and just cut all the nerves. This is the central nervous system. But you've had to cut all of these nerves that are issued from the central nervous system that control the whole body for all the functions. And the peripheral nervous system, or PNS, the brain and brainstem, you're going to have 12 pairs of cranial nerves that control function. And the spinal cord, you will have 31 pairs of spinal nerves. And all of those nerves will do all the function of the nervous system because they'll communicate with the central nervous system. And the spinal cord doesn't do much integration except for reflexes, but it can relay information up and down to the brain, which is the main processing center for the human body. So it's kind of hard to kind of simplify the nervous system effectively in one lecture. But the function shown here gives students a basic idea of how the whole thing works. From this easy picture, the example is thirsty. I want to get a drink of water. Okay, it's easy to understand. Now the input for that, the eyeball sees the water, so that input, sometimes the word that's used is afferent. Afferent information is put into the brain. And so sensory input is basically from the PNS to the CNS. Therefore, you integrate that information, you reach out, you grab the drink of water and bring it to your lips, right? So that's motor output. or efferent activity, affect, effect, afferent, efferent. Afferent is going into you, efferent is coming out of you, a command from the central nervous system. So that information is from CNS to PNS.
You said that was going, afferent is going into and, what did you say?
Yeah, afferent going in, efferent going out. That's what I said.
Thank you.
Okay, so the motor output is from CNS to PNS, basically. That's what I want you to see there. So I just want you to learn motor and sensory, what's meant by that. Okay, how is the information traveling? To give another example, this is kind of like a reflex response, but instead of the brain, they just show a slice of the spinal cord, a reflex organ. And well, I think from this figure you can learn you have sets of cells for the nervous system that have different functions. There are sets of cells for, well, number one, a receptor. In the example, it's a pinprick. So you have sets of cells that act as receptors. It's kind of like the homeostasis thing. You have to kind of detect the sensation, in this case, pain, in the example. That detects it. You have cells for that, and well, that same cell that's detecting the pain acts as the sensory neuron. Okay, so you have sets of cells that have a sensory function. Notice that the information, there's no arrows, but the arrow should be drawn from the needle towards the spinal cord. Okay. Think of the sensory neuron, it's like a telephone cable. It's transmitting electrical signals to the central nervous system. The information is going to CNS. Okay, these electrical signals called action potential. Well, we'll talk about it. It's sensory function. You got to take it to central nervous system. So you notice you have sets of cells that are for integration. Now for integration, notice that the whole cell, that neuron, is called an interneuron. And all parts of that cell are within the central nervous system. Whole cell is within the CNS. Okay, so notice it's receiving input from the sensory cell, the blue cell, and notice the color of the next cell. The interneuron is green. What color is the motor cell? Red. Now blue and red, typically those are the colors for blue sensory, red motor. You'll see that a lot. And notice where the cell body is for the motor neuron. So you have sets of cells that are for motor function. The cell body is in the central nervous system, but then again, like a telephone cable, it sends out an axon to the effector. Like telephone cables again. You have to leave the central nervous system. You have to go to the effector. You're trying to stimulate the effector. I'll write that. The effector could be anything. It could be the gland, it could be, in this case, it's a muscle. So you have sets of cells that respond. Effectors are effected by motor commands. So that's the central nervous system. You have any questions about that? There's like function, there's organization. Basically, all you know now is there's a CNS and a PNS. You know what afferent and efferent is, I think. And you have sets of cells for these different kinds of functions. Like, for example, ultimately, your central nervous system works is if you flex away from the pain, whatever you stuck yourself with. Okay, but you know, the neuron, I have to start with now, like, what is the structure of this cell? Because it's highly variable. So we start with this one here, this neuron cell structure. We had a whole chapter on cells, but neurons have a lot of different specialized processes that really don't come up in a basic cell chapter. Okay, so what do we see here? I mean, neuron cell structure. Now, I already told you there's like sensory, there's receptor, there's integrator. This is a motor cell. This is a motor neuron. Let's start with that since we just had a test on motor units. And what do we see here? Here's the cell body. Okay. Sometimes that's called the Soma. And the Soma has these specialized inputs. I'll just try and draw them like that. They're called dendrites. Now, dendrites have all of these ACH receptors. Call it number 1, dendrites. Think of them as kind of an area of input. Meaning they can receive neurotransmitter signals from other cells. Maybe there's another cell. That's the axon terminal from another cell inputting onto this cell that I'm talking about. Okay?
Instead it receives.
But it's just receiving input from other cells, basically. Yeah. Now, you can, if you're another cell and you want to talk to this cell, you can input on the dendrites. You can also input right on the cell body. So dendrites are an area of input, maybe the cell body, you can input right there, or maybe the soma, another area of input. You can also input right here. It's called the axon hillock. It's right before the axon. I didn't draw the axon yet. Input right there. Axon hillock, another area of input. So you're trying to stimulate the cell to fire. If it does, you need an area of output so it can transmit the signal to the next cell. So the area of output is the axon. And so if you're able to stimulate the cell to fire, the information, it's kind of like, it kind of like builds up, it kind of goes, and it fires down the axon, down this way. So the axon is an area of output. And the signal ends at these axon terminals. So it's like, to get this cell to fire, all of these three inputs had to like stimulate it with neurotransmitters, whatever it may be, ACH or something like that. Stimulate right there. And if it fired, then it would release its own neurotransmitter on the next cell, whatever it is. I'm not going to draw the next cell. But so what you see is you have areas of input, areas of output. You also have other things drawn here. You have these myelinations by the Schwann cells. So each one of these little tubes there is a cell, the Schwann cell. And it wraps around the parts of the axon. Myelination. By Schwann cell. Well, I'll get into that more later, but it helps speed up the nerve impulse. Speed it up. That's what it does. So technically, I didn't just draw the motor neuron. I drew all the Schwann cells that myelinate the axon. So they myelinate the area of output, but nothing else. And basically, there's one little thing, and they point to the space in between the Schwann cells. It's called the node of Ranvier. Node of Ranvier. And basically what that does is you're forcing the nerve impulse to skip to the next node. So it's like you're in action potential, you're generated here, so I'll just spike. And it's like you go to there and you have to like skip to the next one, skip to the next one, skip to the next one. And by skipping, I guess that's like quicker for the nerve impulse, all the way to the end. All the way down here. You just skip all the way, it speeds up the nerve impulse. So myelination, I want to note that.
Speeds up the impulse.
Yeah, increases, well, I guess the fancy way they say it is, you could say increase conduction velocity. Of nerve impulse.
In other words, it speeds up neuron output.
Yeah, it does do that ultimately, yes. Now I put this picture in there because, well, what I just taught you, this cell, it's the first one. It's the motor neuron, right? See this one over here. That red one is this. Notice the cell body here. It's in the central nervous system, right? It's in the ventral aspect of the gray one. It's the right like a telephone cable, it sends out a nerve to the muscle. So that's we call this multipolar because there's many poles. One is the axon, some are the dendrites. So if you want to put a definition to this structure, it's multipolar. Some sensory neurons I want to define. Sensory neurons are different from motor neurons. They really don't have those dendrites. I usually don't see no dendrites. Where dendrites are area of input or output. It was the input, very good, yeah. Now here's the thing, just because they don't have dendrites doesn't mean they don't have an area of input. Remember, neurons are highly variable in their structure, but all neurons have an area of input or output. They just may not have dendrites. All neurons. Have areas of input and output. In the case of sensory neurons, well, they don't have dendrites. For example, what's a bipolar node? You usually see those in a stack of cells in the retina of the eyeball to convey the sense of vision. Well, anyways, you'll get there later. But so what you're seeing there is like you have this kind of bipolar structure, two poles. It's in the retina. Now it has an area of input or output, and just drawing it on the board like that, you can't tell which is which. So I'll just arbitrarily point to one end. Let's say that ends an area of input. That ends an area of output. It's just there's no dendrites, no but there's area of input and output is the point. It's a different structure. One over here. This is the one we did for the skin. So it's like you have free nerve endings here. So let me just kind of draw it like this on the board. It's called unipolar. Okay, here's the cell body. It's right there. It has one pole, unipolar. It's very short, like that. But then it immediately divides into two parts. It goes here. It goes here. Now let me draw one longer than the other. Okay, and here's the thing. The longer one is out in the periphery, in the body. Okay, like in the picture there, the longer end is the end that's in the skin, right? So that's the area of input. It's kind of sensing pain, temperature, touch, all those different things. Input's right there. And that process, the longer process after it divides into two, is the peripheral process. In the shorter process, the area, I guess, of output, the one going to the central nervous system, is the central process. So, in terms of function, bipolar cell seeing unipolar cell feeling, seeing and feeling, is that sensory or motor in its function? Sensory or motor? See and feel. Sensory, right? It's not hard. So unipolar, they're sensory. These are sensory. Notice where the cell bodies are. The cell body right here, it's in the retina. Look at the picture. Is the cell body, is it inside or outside the spinal cord? It's outside. Now I'm going to point to it there. It's a little dot on the screen. It's outside the central nervous system. It's right there. We'll get to that with the spinal cord. It's called the dorsal root ganglion. So I always tell students, pay attention to where the cell bodies are. If you damage nerves, if you damage the processes, it's possible they could regenerate. If you damage the cell bodies, they can't regenerate, usually. That function's gone. So it's kind of important to know where the cell body is. If you squash a nerve and make your arm lose function for a while, if the cell bodies are in the central nervous system, they can regenerate the broken processes. It might take several months, but with spinal cord injury, a lot of times you lose that function forever. Maybe you're paralyzed because you've lost the motor function. That's really important to think about. Okay, one more cell type before I move on. I didn't have a picture of it there. You even have, okay, so for these cells, there's no dendrites. But you got axons. There's just no dendrites. You even have hair cells. I guess I'll draw it sideways. All these little hairs. This is this little hair bundle. The hair bundle is the sensory apparatus. That's the area of input. So I guess the area of output is on this side. Here's the thing though. There's no axons or dendrites in the hair cell. No axon, no dendrites. You still have areas input and output. The hair cell is very important for auditory and equilibrium, hearing and balance. These guys. So to recap, motor neurons, they got input and output, the dendrites and the axon, and sensory. No dendrites, but they have input-output still. Sometimes they have nothing, just input-output, but no axons or dendrites. I put a picture of the brain next to a functional picture where it has kind of cells, sensory and function going up. The black ones are interneurons and red is going down. So I want you to know that when people use the term nerve pathways, you have multiple cells in a row communicating with each other. That's kind of the concept there. A number of cells in series. That's kind of how it's described. Series means one after the other, just like the picture. So look at the picture. Look at the blue cells. Is information ascending or descending to the brain? for sensory. If you look at the arrow here, that's a clue. Information is ascending to this cell, and then to that cell, and then to that cell. If you're going to feel something, it has to reach your cerebral cortex. Otherwise, you don't feel it, right? But then you have integration, the black cells, interneurons, and then the motor pathway. Is the information going to ascend or descend? Well, it can't ascend anymore on top of your brain, so information descends. Okay. So notice in the spinal cord, in this example that's given, you have two information pathways. One is ascending, one is descending. So information can be sent through multiple chains, all right? So you have one nerve pathway, that's a series, but if you have multiple nerve pathways, what they say is information can be sent through multiple chains. That's called, it's parallel, right? I mean, for example, in this simple one, if you have one going up, one going down, they're parallel to each other. The information is going in different directions. But those are two nerve pathways in parallel, one's motor, one's sensory. That's the idea of a nerve pathway, just cells in series, because cells have to talk to each other, and that's kind of what I have here, this concept of a nerve pathway, that cells are lined up in a row. And if you just look at the simple picture, let's count the number of cells it takes to reach the primary sensory cortex. That's sensory, that's ascending. How many cells, how many neurons does it take to reach up here? Let's count. Here's the first cell, the receptor. 1, 2, 3. It only took 3 cells to get there. Okay. Then you have the interneuron. Now count how many cells to get down. 1, upper motor neuron. 2, lower motor neuron. So it only took 3 to get up, 2 to get down. That's it. So it's not like a million cells. Just 3 or 2. That's it. So sensory information is sent through chain of 3 cells. Okay, the direction is it's ascending to the cortex. So for motor, is sent through a chain of 2 cells, but the information is descending. So that's the basic idea. All right, now you have all this information, ascending or descending. How do we categorize the information? So this figure is good to study. Okay, it's kind of hard to teach from because the font's really small, but it has all the different kinds of motor and sensory functions we expect students to know. So on the left, it's the central nervous system. You see brain and spinal cord, and they have all these lines going to and from it, because the central nervous system, what it says in the gray box, is communicating with motor and sensory, the peripheral nervous system, cranial nerves and spinal nerves. Okay, so the arrows are going back and forth. And if you see a blue box or a blue arrow, what function is that? It's always sensory. And if the information's coming down, and red lines, red boxes, it's motor, it's function. So, okay, let me kind of define these different terms. Let's start with the sensory division. There's different kinds of sensations that we categorize. Sensory or afferent division. Basically, there's somatic and visceral sensory, those kinds of sensations. Soma is the body. Somatic sensory is basically the sensation of touch. Your skin. Your skin is basically one big touch receptor. I also kind of throw in with this category the idea of proprioception. And that's kind of like, I always give the example of, have you ever had the sensation of your arm falls asleep, you slept funny, and you wake up and you don't feel your right arm. It's very disconcerting, but if you just kind of shake your arm, blood flow returns and you can feel it again. But anyways, that momentary loss of sensation, you can't feel your arm, is proprioception. Information from your joints and muscles constantly communicating to your central nervous system. Yes, I'm aware of my body limb. When it's not there, that's kind of the only time you appreciate proprioception. Well, anyways, that's somatic sensory. Visceral sensory, describe it. Visceral means contents, like your organs. Like, for example, you feel indigestion. You eat bran muffin and coffee, and there's a gastrocolic reflex. You feel like you gotta go. Those feelings are visceral sensory, okay? And you feel the digestion, cramps, things from your gut, visceral. So it's all about touch and feel. It's all sensory, you feel it. So any questions on sensory? What's visceral, what's somatic? So when those terms are used, you're okay. All right, now, motor. I have a little more categories. Motor function, efferent division. Motor can either be somatic or autonomic in its function. So I'm going to pull it up here a little bit. Motor function, if it's somatic motor. That's information that's descending, that's going to control skeletal muscle. Okay. It says mobilizes. It says this is voluntary, conducts impulses to skeletal muscles. Because remember, skeletal muscle is your voluntary muscle. And you did a lab on EMG with the whole clenching, right? And it's the effort, and you can see it on the EMG. That's somatic motor. Pretty much everything else is autonomic. Now your autonomic nervous system has two divisions. There's a sympathetic and a parasympathetic, and they kind of counteract each other. So when you're autonomic, you have to consider, well, is it sympathetic or is it parasympathetic? When I say they counterbalance each other, let me give you an example. If I say the sympathetic effect is to increase the heart rate, what would the parasympathetic effect be? To decrease it, right? The sympathetic effect is to increase blood pressure. The parasympathetic will be to decrease it. So you have this kind of, they kind of counteract each other kind of a thing. Autonomic things will basically control organs that have smooth muscle. And also organs that have glands. I think examples could be like, not the thyroid. The thyroid is controlled by other things, hormones. Like sweat glands, tear glands, mucus glands, those kind of glands. Sweat, tear. Sweat, tear, mucus. So those kind of, that. So we call it visceral motor, but it could be parasympathetic or sympathetic. So that's the thing you gotta keep straight in your head. So you're hungry, you see someone else's food, you start salivating. That's visceral motor, okay? You get your exam back, oh man, it's not the grade I wanted. You start crying, it's visceral motor. Or it's the grade you wanted, and you start crying, visceral motor. You know, gland stimulation is a visceral motor function, is basically what I'm trying to say. Sympathetic, parasympathetic, give you a couple examples there. They say, conserves energy. They say this one mobilizes body during activity. They call this the fight or flight response. And they call this one the rest and digest response. So imagine like all the things that happen after you digest a large meal, how you feel. That's the parasympathetic taking over. Fight or flight is literally that. It's a when you're startled. Most people react by crouching away in fear. Some people react by punching them. That's true. I mean, you can put a hidden camera, have someone pop in the box and people are walking by. Some people, maybe 1 out of 10, will just punch that person in the face as a reflex because they fight. That's their response. Some people, most people are just kind of just get scared and crouch away. That's flight. We basically use the sympathetic in extreme stress. Okay, like a small child is trapped in the car and grandma lifts up the car. Grandma's not strong, but all the catecholamines from a sympathetic response give her an increase in strength metabolically for that moment. So those are two divisions of autonomic. So now, having defined all those terms, the terms you got to keep straight in your head. First ask yourself, in my examples, is it motor or is it sensory? Is it afferent or is it efferent? Well, if it's motor, which one is it? It's efferent. Okay, all right then, but then is it somatic or autonomic? So you have to read the example. Also, if it's autonomic, is it sympathetic or parasympathetic, okay? And then so that kind of tells you if it's somatic or visceral. So what I like to do is, I have that Cahoots quiz. Remember we did Cahoots? Let's do it again. This time put your real name, just so I can see that you did it. Don't put a nickname. I want to know that you were here and you did it. If you're not here, I mean, those people aren't going to lose credit, but for my own records, I like to see who did it. You're not going to gain or lose credit, though.
Not a full name.
Something I can recognize, maybe first name, last initial. It's good enough. So we do classic. Start. And it gives you the pin. You just go to, on your browser, on your device, after I go through this. So if you remember how we do this, you just kind of go to Kahoot.it on your browser and enter the game pin and I'll start. So put a name I can recognize you by, not just a nickname. pretty good. We'll get started. Join anytime if you're not joining yet. So I'm giving you examples. I want you to categorize it. A neuron carrying signals away from CNS to biceps brachii, which is a muscle, causing it to contract. Which of those would you choose? The choice to say somatic sensory, visceral sensory, somatic motor, visceral motor. It's okay. Join any time. You're fine. Those are that answer, kind of all over the place. It's somatic motor. Let's revisit this. It's a neuron. It carries signals away from CNS to biceps. So if information is going away from the CNS, is that motor or sensory?
Motor.
That's motor. So that's the first clue. It's motor. And it's going to biceps brachii, which is muscle. Maybe you don't know that yet. Your biceps is a muscle. If it's muscle, it's somatic motor. And for those of you who responded, most of you got it. So let's keep going. These quizzes are public. If you do terrible right now, after you study, do it again, maybe you'll do better. The neuron carrying signals away from CNS to the heart, increasing heart rate. How would you categorize that? It is visceral motor. Most people did put that. Okay, it's going to the heart. Heart's an organ. That's a viscera. I tell you, it's increasing the heart rate. Okay, that's visceral motor. Most people got it. Okay, let's keep going. Yes.
What is it behind it?
Huh? Oh, it's usually osmolar. Sorry. Not gonna remember that. Thank you.
Are you able to turn the light off on top because I can't see?
Oh yeah, let's do that. Maybe that'll help. Yeah, you're right. It doesn't show very well. Just oops. All right, so let me read this one. You feel a hot candle burning your skin. Somatic sensory is correct. Okay. Your skin is your biggest touch organ. Skin is somatic, basically. Avoid your bladder is, number one, you urinate. Okay. It's visceral. Well, yeah, I guess you wouldn't know that. It's visceral motor. Okay. But I guess you wouldn't know if it was sympathetic or okay. Well, most of you put sensory. When you void your bladder, that's not sensory. It's a motor thing. You're instructed pee in a cup. That's a motor function. All right. So that's why visceral sensory cannot be correct. Although you probably do feel that your bladder is full. That's sensory. But I didn't say that. I said void your bladder. That is a motor command. So that's why visceral motor parasympathetic is correct. feel the bran muffin you ate digest in your stomach. You feel. You feel.
Yeah. Keyword feel. Yeah.
Well, to determine motor or sensory, yes. All right, well, that should be, oh yeah, visceral sensory, and most people got that. All right, well, oh, random one, okay, great. But anyways, again, not keeping score, but take that quiz again. It's public, and maybe it'll do better the next time. So I'm just trying to get you to think about the different sensations and motor functions for the nervous system here. You didn't do too bad. Those examples are right here on the PowerPoint slide too.
Oh.
You feel a hot candle burning your skin, you void your bladder, feel the bran muffin in your stomach. All right, so I want to take a step back from function and look at the anatomy here. We'll take a look at this picture. It's really histology. You have gray and white matter. Okay. You can see this without a microscope, by the way. Gray matter has a lot of cell bodies in it. What makes it gray? The white matter is usually due from the myelination. Myelination is fatty and makes it whitish. So in nervous tissue, I already talked about the neurons. The neuron is the main kind of cell that we're concerned with. Oh, my bullet points got a little messed up. So you have gray and white matter, great. Now the cell types for the histology I would like you to know. I already talked a lot about neurons. These are the main cells of the central nervous system. They're not the most numerous though. The glial cells are. But the neurons, they're the excitable cells that can generate electrical signals. Not so the other cells. The glial cells, they can't generate electricity at all. I'll just say that. Can't generate electricity. Basically, they have a supportive role. They support the neurons. Here is just a list of the ones I think you ought to know, and I'll go through them. There's nice pictures here that we can look at. The different types of glial cells, and before I go through them, I should note, these kind of like outnumber the neurons like 10 to 1. They're like way more numerous. But we kind of give them less attention because it's really about the physiology of the neuron that's important in terms of physiology, but I'll give mention to the basic function of some glial cells. So here are Astro Sites. Okay, they have paravascular feet. Oh, why is it getting cut off there? Let me restart my PowerPoint app. Maybe it'll. Sometimes when I restarted it, doesn't cut the slide off anymore. Let me just try that. That's better. So Astro Sites with paravascular feet. So those processes, do you see how the paravascular feet wrap capillaries? And what you're doing is, capillaries are blood vessels that allow exchange of metabolites. And when you wrap around the capillary, don't you see how you limit the exchange? And that's what you want. In the brain, the filtration of the blood is very important. We call it the blood-brain barrier. That's the barrier. It's like those paravascular feet limiting the capillary exchange and warm blood. They also help recycle neurotransmitters. That's another function of the astrocytes. Help recycle neurotransmitters. Moving on, microglial cells, they function like macrophages. They kind of phagocytize microorganisms. All right, next up we have these ependymal cells. They're basically epithelial cells. They look like a columnar epithelium. Their function is to form an epithelial lining. They help generate CSF. Form a barrier between blood and CSF. Okay, so it's called the blood CSF barrier. So you have the blood-brain barrier, not to be confused with blood CSF barrier. I think of it as your nervous system, the tissue should not be exposed to whole blood. It's kind of poisonous to the nervous system. And so it's like, if this is like your brain and your spinal cord, your cerebellum. I'll get into the meningeal layers later, but there's connective tissue layers all over your central nervous system. This is the red one. It's called the pia mater. Blue layer around it. It's called the subarachnoid mater. And outside of that is the toughest one, the toughest connective tissue covering around the whole CNS. It's called the dura mater. Which means tough mother. Now the reason why I mention that is when blood is filtered through these ependymal cells at particular locations and you form CSF, the cerebral spinal fluid is basically blood. It's like clear fluid. Okay, it's filtered blood. And it circulates around the whole central nervous system, but it does it underneath the subarachnoid mater. So it's kind of like circulating at this level here, between the red and the blue, the circulates. Okay, I'm simplifying it, but CSF circulates. So CSF circulates at the, you're bathing the blood in, excuse me, you're bathing the brain in CSF, not whole blood. So that's the blood-CSF barrier. The ependymal cells help create it. So the blood-brain barrier, you're kind of talking like the space between neurons. That's called the interstitium. So the blood-brain barrier is guarding the CNS interstitium from whole blood. Okay, this barrier is like you're creating a barrier between blood and you're creating CSF. So not only are you a barrier between blood and CSF, the ependymal cells actually help create it. I should put that word too. Anyways, I just wanted to mention ependymal cells. They help form the CSF. They help be a barrier. So on that figure, where it says fluid-filled cavity, it ought to say CSF. Okay, and it's circulating it throughout the whole central nervous system.
How does the ependymal cells regenerate the CSF?
But, okay. Basically, by being close together, there's tight junctions between them. So what they select to let through is how they create it. Only very small molecules like glucose and oxygen. In other places of the central nervous system where you have ependymal cells, they don't have the gap junctions and a lot of things can get through. But only in places where you have these cells that have the tight junctions, that generates the CSF. It's just the tight barrier that they create. The next type of glial cell is an oligodendrocyte. What I want you to know about them is the cell is the blue phase. Notice how the process wraps the nerve fibers? That's myelination. So these myelinate axons within the CNS. And basically, those cells, yeah, I already said what I want to say, myelinates, axons of neurons in the central moving on. This picture shows Schwann cells and satellite cells. The Schwann cells, I'll do those first, since they have similar functions. The myelinated axons of neurons in the PNS. I already mentioned that. You got the node of Ranvier, right? What also pictured around the cell body are satellite cells. Satellite cells are called that because like it's like they're orbiting the cell body or something. They function like astrocytes. They help recycle neurotransmitter. Here's a picture of a Schwann cell wrapping an axon. So you can see that it's kind of many wrappings of cell membrane around an axon, which is a part of a cell. So if you look at it in cross-section, you can see the diameter of the axon, but at the top of the figure it shows you the thickness of the myelin sheath. That's where you force the nerve impulse to skip over that. Because basically, the myelin sheath, if you think about what a cell membrane is, it's fat. Fat cannot conduct electrical signal, so when electricity gets to it, it has to like jump over it. Okay, well anyways, that kind of gets us to the next topic, membrane potential. That's the end of the glial cells. Talk about the neurons now. Now the resting membrane potential, going back to neurons, neurons are the excitable cells. So what if you're not excited? So I guess you could be resting. Or you could be in a state of responding, you're stimulated. The membrane potential is the voltage difference in charge across a cell membrane. Okay, forget the word resting right now. Let's just focus on membrane potential. That word, potential, that's the key word. Membrane potential is the difference in voltage across the cell membrane. So that resting membrane potential is the membrane potential measured during when the cell is not excited, when the cell is not stimulated. Resting membrane potential the voltage measured in a neuron in the absence of stimulus. So they give you a simple picture here to help understand this. I guess before I move on, I should define some basics physics terms. When you take kind of freshman physics, what we call it, they teach you, I think it's in like the second semester or third quarter, Ohm's law, which is voltage is really, you're measuring current, it's a product of the resistance, V equals IR. Okay, so now we're getting into electricity, and if you don't know what the electricity is, you don't have to know much. Okay, just conceptually, just look at the picture. I'll just tell you that cell is not stimulated, so therefore it's resting. And look at the number there. They actually measure a difference, a charge difference between the inside and the outside of the cell. And notice where they're taking the measurement, that they're choosing the axon, right? So what voltage is, when people say that, I think we use the units volts, but it's actually a person. You can look up what the units mean, but we use units of volts. It's a separation of charge between two locations. So I guess I'll just kind of draw top of their thing. Here's my cell membrane. And it looks like they have positive charge on the outside, and they have negative charge on the inside. So those charges are separated, okay, by the cell membrane. You know, this is not like an equilibrium thing. This is the condition of rest, where you have this charge separation. And if you take your electrode and you're able to get it like inside of the cell membrane versus outside, I mean, you're going to measure that difference because you can't see it. You use a voltmeter. The inside is negative with respect to the outside, so that's why they measure the resting membrane potential is negative 70. Negative 70. So the negative means with respect to the inside. That's the orientation, with respect to the inside. The inside is more negative than the outside in the condition of rest. And it's different for different mammals, but negative 70 is a typical textbook number. So that's the condition of rest. Other things, current and resistance. Okay, current, I don't know why they use the symbol I. I in physics is current. For us, it's biological current is ions flowing. Ions moving, that's current. Now, how can ions move? Well, I guess if you had a channel, if ions move across a cell membrane, need a channel for that. If ions move, I don't know, say in. If ions move through a cell membrane, that's current, okay? Because it changes the membrane potential. Ions can so ions flowing into a cell, that's current. What if these positives get in and they see everywhere else it's negative, and they want to go towards negative because positive is attracted to negative. So this little gang of positive cations, they move towards negative. That's current too. So it's like you can have flow of ions along the inside of a membrane. Okay, that's current. So that's voltage, the separation, the current flows. The R is the resistant flow. Okay. And the resistance to ions flowing is the cell membrane in this case. Because the cell membrane has this hydrophobic zone where charged things can't get through. So, cell membrane. So that's basically my description of voltage, current, and resistance. I usually don't talk about resistance because it's a constant. It's always the cell membrane. Any questions on voltage or current though? The definitions? I want to talk about the establishment of the resting membrane potential. This negative 70. Cells create a very uneven situation for their rest condition. Students like to mistakenly call this equilibrium. Now why is that a mistake? It's a common mistake. There's nothing equilibrium about this. If this were equilibrium, what should this read? 0. As much positive as negative, inside versus outside. You measure no difference. That would be equilibrium. Okay, but this is not the case. It's negative with respect to the inside. So I want to talk about how cells establish that. Let me read what I have on the slide. I say that a neuron at rest has an excess of positive charge on the outside and negative charge on the inside. The rest is negative 70. And it's largely established by many leak channels. Okay, that's an important point. And then I say, establishing this is as easy as 1, 2, 3, ABC. So I'll explain what I mean by 1, 2, 3, ABC. But this kind of emphasizes something we already talked about in chapter 3. I don't know if you remember studying the sodium potassium ATPase pump. This is that. This is basically that. So I teach it in the first test, so when it comes back to this test, it's a little easier to understand. So let's say you have a cell membrane. I'm going to draw leak channels. These are going to be potassium leak channels, the red ones. And I'm going to draw more of the potassium leak channels than the sodium leak channels. And then between them, I'm going to put the ATPase pump. And let's review what we talked about in that previous chapter, the first test. It was a while ago, so maybe you've forgotten by now. As I've drawn it, I'll just designate this part of the cell to be inside the cell. Now, you already know it's negative with respect to the outside. Something like negative 70, if you were to measure it. Now, part of what makes it negative is there's a lot of negatively charged proteins in the cell. Proteins usually have a net negative charge that contributes to the negativity, but mostly it's largely also due to this the leak of sodium and potassium. Red is potassium, green is sodium. So we say permeability. So what I'm going to write is that the cell is permeable to the potassium and the sodium. That's what I'm going to say. Students don't like the word permeable. It just means it can leak in or leak out. Because if those leak channels aren't there, the sodium or the potassium, these are both leak. Call it, I would write leak, so you make a connection. Leak channels are these. If the leak channels weren't there, sodium and potassium could not get past the cell membrane, and the cell would not be permeable to it. Now these ions help establish the negative 70. That's why they're important to mention. And I say on the slide, the resting membrane potential is established by many potassium leak channels. I don't even mention the sodium, but they're there. That's why I drew more potassium leak channels. And if you remember from that previous lecture, there's more potassium inside the cell than outside. So the concentration of potassium is larger inside than outside. So I'll put a smaller K outside. And the concentration gradient for sodium is reverse. There's more sodium outside than inside. That's what I taught you before. More sodium outside compared to inside. So these are concentration gradients. However, because these both carry a full positive charge, we also have to consider the electrical gradient too. So what I would write down is consider the electrochemical gradient both. So consider the electrical chemical gradients for sodium and potassium. Just make sure I'm both here. Okay, so you have your leak channels and they're open, and so you have a gradient, concentration gradient for potassium. And what do we say? Will potassium leak in or out based on the concentration gradients? It'll leak out. That makes you have more here, less there. You flow, if you can flow, from high to low. So potassium will leak out. And let's just kind of stop there and think about what's happening. I'm going to write on this board here. So what's happening as potassium leaks out, think about it per ion. For every potassium that leaks out, you leave one excess negative charge inside because one positive is leaving. Kind of by default, that's what you say. You leave one excess negative, that's a negative sign, not a dash, one negative charge inside. Do you see that? Positive is leaving. That's making the inside more negative. Because a positive is left, and therefore by default, they're more negative. So therefore, that excess negative charge, it kind of wants to pull potassium back in. That's kind of like the electrical difference there. The excess negative, excess. Wants to pull potassium back in. So just talking about potassium, it's like it wants to leave due to the concentration gradient, but as it's leaving, the excess negatives want to pull it back in as it's trying to leave. So we call that, if you just let potassium do that, if you just let potassium flow until the concentration gradient, the force of that exactly equals the electrical gradient, that's called the equilibrium potential for potassium. So let me write that. If you let just potassium, if you let potassium flow until the potassium concentration gradient equals the potassium electrical gradient, and you measure the equilibrium potential. The equilibrium potential for potassium will be negative 90. That's kind of the thing to note there. So, at this negativity, it's so negative inside the cell, positive potassium can't leave anymore. So that we call that equilibrium. It's just not going to leak anymore at this one. Okay, I want us to have the same conversation for sodium, because it's also leaking. So what I'll write is for every sodium that enters, you get one excess negative on the outside. For every sodium that enters, you leave one excess negative charge outside. So I'll draw a sodium leaking in here. Okay. It's the same thing. It's like a positive going to the inside. You're leaving excess negative on the outside. So think of it the same way. That excess wants to pull sodium back out. If you let sodium flow till it reaches its equilibrium potential, right, the same thing. If you let sodium flow until the sodium concentration gradient equals the sodium electrical gradient. Well, basically, the equilibrium potential for sodium will be about plus 66. So what that means is the inside is positive with respect to the outside. It's so positive in here, plus 66. Positive tries to come in and it can't. It's too negative out here. It's like you're pulling it back out. So that's kind of like, you see how far away they are, these equilibrium potentials. What was the resting membrane potential? It wasn't either of these. What was it? It was negative 70. So what is that closer to? It's closer to this. So what that tells you is the cell is more permeable to potassium. So here's how I kind of sum it up, everything I talked about so far. Let's see. Then I'll mention the sodium potassium ATP stuff. So I'm going to erase that. So I established all the ground rules to understand how you get that negative sender. We talked about the leak, we talked about, well, we didn't really talk about the pump yet, but you know what it does, so we'll kind of throw it in the mixer. But the first point you should take away here. So to maintain that negative 70, that's our RMP, our resting membrane potential, you need to maintain the ionic gradients that you have for sodium and potassium, okay? Cell must maintain ionic gradients. You need the leak channels there to be permeable to these things, but you can't let the leak channels destroy the ionic gradients, okay? So that's my second point. The passive leak of sodium and potassium would destroy these gradients. So the third point is the pump. The pump stabilizes the negative 70 by maintaining the gradients of sodium and potassium, which we already learned. Okay, we already learned about the sodium potassium pump. That's my third point here. The sodium, potassium, ATPase pump stabilizes our resting membrane potential, our minus 70, by maintaining the ionic gradients. So let's kind of draw what that is. That'll be my first point. Let's call it A. So 3A. Potassium leaks out, sodium leaks in. That's what we already said there. Oh, I'm sorry. I didn't want to put B next to that. I just wanted A to be potassium leaks out, sodium leaks in. Okay, so you already wrote that. For B, put what the pump does. It recaptures the 2 sodiums and ejects the 3, it recaptures the 2 potassiums, ejects the 3 sodiums. Let me write it correctly and draw it correctly. Okay, so B, the pump recaptures 2 potassium ions. The pump will then eject 3 sodium ions. That's kind of the 3-2 ratio we discussed before. So let me illustrate that. If potassium is leaking out, the pump kind of pumps them back in against their gradient. Potassium, potassium. Okay, and then the opposite is true. It'll take 3 sodiums that are leaking in and pump it back out against the gradient. Sodium, sodium, sodium. That's the 2-to-3 ratio. And you expend ATP to do that. That's the other thing we talked about in the previous chapter. There's an ATPase. It takes ATP, you kind of break it apart into ADP plus inorganic phosphate. So that's how the pump works. And what you're doing is you're counteracting the leak. So we write that as my third point, C. The active pumping exactly counteracts the passive leak. Well, unlike keywords, active and passive. The leak channels are passive, but pumping is active. I use the same word up here for passive because passive means you're not expending, the cell is not expending ATP. Leak channels just don't expend ATP. The ions just flow down their concentration gradients to a point, but active is we need to expend ATP to maintain the gradients. So there I kind of describe everything we talked about here, and I remind you of how the pump works for A, B, and C. So again, what's the end result? What's the number I should write after C? Yeah, -70. It's close to -90 because the cell is more permeable to potassium, so it kind of like lives closer to its equilibrium potential. I want to look at some slides that will kind of help you solidify all of these concepts. So that's just a picture of the pump to remind students of the 2 sodiums being recaptured and the 2 potassiums being recaptured, the 3 sodiums being ejected. This slide is pretty good. Look at the top figure there. I'm trying to show you the electrical chemical gradient thing that I went over. And notice that there's more potassium inside than outside, and you measure the difference, and it's negative 70. Notice the gradient for the chemical gradient, the concentration gradient is stronger than this one, the electrical gradient. So the net gradient is out. Potassium leaks out. Now, what I was saying here is if you just let it flow until both forces are equal, that's the bottom figure.
Okay.
So what they do is they say, okay, what if you just let potassium flow until all the negative charges in here want to pull it back in so that this force is exactly equal to that one. And then you measure it. It's negative 90. Okay, so these forces are equal, so it wouldn't flow anymore. Okay, now that's the point of that slide. And they do the same thing for, put a similar slide in for sodium here. So I'll just say, I'll stand over here. Look, it's, all the arrows are pointing in. Sodium leaks in is the chemical gradient. The electrical gradient is pointed in too, because it's negative on the inside. So if you're positive, you're attracted to a negative interior. Okay, so sodium leaks in. But what I was saying, if you just let sodium flow until the inside is so positive, then it can't do it anymore. It's so positive, it's plus 66, therefore they flip the arrow on the electrical gradient. It's like it can't go in anymore. This is the equilibrium potential for sodium. Okay, so what I was saying is the action of the pump keeps you at negative 70 to counteract the leaks so you don't reach equilibrium. I guess I like this figure the best. It's the most simplest one. They kind of included everything I did. They have proteins with negative charges contributing to the negative 70. Here's leak channel. Are those active or passive?
Passive.
The passive, and the pump is the active. Yeah, and it's kind of like leaking in. Oh, okay. I'm sorry, leaking out, get it back in. This one is leaking in, eject it. So it's all right there. It's really easy to remember. Just look at that one picture. The Marriott book, sometimes they list different figures to try to help students understand. So I included them as part of my PowerPoints. This one here is giving you the scenario, what if the cell was only permeable to potassium leak channels? Then that's why they have a negative 90 there. Now if you introduce a sodium and you get some positive in the cell, it goes up to negative 70. And if you get a pump in there, it'll maintain the leak at 70. So that's kind of how they try to teach the concept. This contingent of rest is not equilibrium. It's leak channels and it's the sodium pump working together. Now I can't get this to work, so I'm not going to try to play it. I think I explained enough. And I want to stop here. I wanted to leave a little time because your EMG lab is due today. So if your group finished it, just turn it in, you're free to go. But if you need to finish it, now's the time. And I'll see you Wednesday.